Okay, hello you guys. In this video, we're going to go ahead and move on in the 5.1 notes. In this video, we're going to look at topic 2, which is all about meiosis and how gametes are made. And so if you guys recall from the last video in topic 1, we were talking about sexual reproduction and how in sexually reproducing organisms, there's two parents involved in contributing DNA to the offspring. And in order for that to happen, each parent has to make haploid cells that have one set of chromosomes, or half the number of chromosomes as the rest of the cells, the haploid cells. And those are the sex cells, the sperm and the egg cells, which we call gametes. So in order for sexual reproduction to happen, you have to have these parents making gametes, sperm and egg cells, that have half the number of chromosomes, or just one set of chromosomes each. And then those gametes, the sperm and the egg, can fuse together to produce an offspring that's now diploid, who has two sets of chromosomes, or the correct total number of chromosomes. And then that fertilized egg can then start dividing and growing and developing into the offspring. Now, in order to make gametes, to make sperm and egg cells, there's a special process of cell division called meiosis that needs to take place. And so in topic 2, we're going to look at how meiosis takes place and how you end up with these haploid cells that are going to act as your sperm and your egg cells, carrying just one set of chromosomes. And so here's the gist of what's going to take place, and then we'll go into a lot of detail. But the overview of meiosis, basically you're going to start with interphase, which this is just like mitosis so far. You're going to start with interphase. And interphase, what happens is the chromosomes are going to duplicate. So each of the chromosomes are going to make a copy of themselves. The DNA is going to replicate, and you're going to make a copy of each chromosome. And those copies are still going to be held together as one chromosome. And those are called sister chromatids. Sister chromatids are two copies of the same chromosome still held together as one chromosome. And so that's going to happen during interphase. Before you do mitosis, before you do meiosis, you always have to do interphase, which is when those chromosomes have to duplicate. Now, once you have duplicated chromosomes, these chromosomes that have made a copy of themselves, you are now ready to do cell division, which in this case, we're going to do meiosis. And in meiosis, there's actually going to be 2 rounds of cell division. The cell's going to divide once, and then each of those cells is going to divide a second time. So you're actually going to end up with not 2 daughter cells, but a total of 4 daughter cells when meiosis is over. And those 2 rounds of cell division are called Meiosis I and Meiosis II. So Meiosis I is the first time when the cell divides, and Meiosis II is the second time when the cell divides. And then after Meiosis I and Meiosis II, you have now finished meiosis, and you're going to end up with 4 daughter cells, like I said. But each of those daughter cells are going to have just one set of chromosomes, like one of each type of chromosome. They're going to be haploid. And those chromosomes are going to be unduplicated once you end meiosis. And so now we're going to go into the details of what's actually taking place here in meiosis. And I'll try to compare it to what was happening in mitosis, because they are different and there's some important distinctions between them. But let's start with Meiosis I. So the first round of cell division during meiosis. So in Meiosis I, this is when the cell's going to first divide. And what happens in Meiosis I is these familiar phases, prophase, metaphase, anaphase, telophase, just like in mitosis, that's going to basically happen twice. There's going to be prophase, metaphase, anaphase, telophase, and cytokinesis. That's going to be Meiosis I. And then there's going to be another round of prophase, metaphase, anaphase, telophase, and cytokinesis again. That's going to be Meiosis II. And so when we're talking about Meiosis I, we call each of those phases prophase I, metaphase I, anaphase I, and telophase I, because we're referring to the first round of cell division in Meiosis I. And basically what's going to happen in that process, which actually I want to show you visually first before we go into the details. So here's a little pretend example. And so pretend that my table here, my desk, is a cell. And inside this cell, there's 4 chromosomes, right? 1, 2, 3, 4. There's 4 chromosomes. In actuality, there's 2 sets of 2 chromosomes. So we have one of these small chromosomes. Let's say this is from your dad. And you have another small chromosome, and this one's from your mom. And on this chromosome, there's the same set of genes on it. Let's say there's the gene for your skin tone and for your height. So somewhere on this chromosome is the instructions for your skin tone and your height. And your dad gave you a chromosome with those genes on it. Well, your mom also gave you a chromosome with the instructions for your skin tone and your height. Okay, these are homologous chromosomes. They're two different chromosomes that have the same set of genes on them. Now, those genes could have slightly different instructions. They could be different versions of those genes. So for your skin tone, your dad could give you instructions that are different than the instructions your mom is giving you for skin tone. So they're not identical, but they do have the same set of genes on them. Those are homologous chromosomes. And then you have another set of chromosomes, or another chromosome from your dad. This one's larger. Let's say that this chromosome has the instructions for your eye color, your hair color, and your hair type. If you have curly hair, wavy hair, straight hair. And then your mom also gave you a chromosome with the instructions for your eye color, your hair color, and your hair type. And so these are also homologous chromosomes. So this is a pair of homologous chromosomes. This is a pair of homologous chromosomes. Hopefully you guys are all with me so far. So there's 4 chromosomes. So the diploid number in this fake example is 4. In a diploid cell, here's a diploid cell, there's 4 chromosomes, or 2 sets of 2. 2 from your dad, 2 from your mom, which form 2 pairs. This is a homologous pair, and this is a homologous pair, okay? Now, during interphase, just to remind you what happens during interphase, we're going to make a copy. Before you do any type of cell division, before you do mitosis or meiosis, you got to make a copy of each of these chromosomes. They're going to duplicate. And so what that will look like is we're going to take this red one from your dad, and it's going to copy itself. And then those 2 copies are still held together at the centromere. Okay, they're still attached, but these are exact copies of each other. We call these sister chromatids. So this is an exact copy of that, this chromosome duplicated. And that's what's going to happen in interphase. In interphase, all of your chromosomes are going to duplicate. So that one duplicates, this one duplicates, this one duplicates, and this one duplicates. So now you still have 4 chromosomes, but at the end of interphase, once you've replicated and duplicated all of your chromosomes, you now have 4 chromosomes, but each of them are duplicated. So this is 4 duplicated chromosomes, and each chromosome now contains 2 copies of itself, right? Now, when this happens, just to clarify some things for you guys, we still call these homologous chromosomes. Like on this chromosome, this is a specific chromosome, and it has the instructions for your eye color, your hair color, and your hair type. But now we just duplicated it. And there's a second copy of all that information. But this other chromosome, that's another chromosome, a separate chromosome from your other parent who also has the instructions for your eye color, your hair color, and your hair type. And like these are still homologous chromosomes. The only difference now is now they've duplicated. Okay, just accept that that's true. Like this is still 2 chromosomes that have the same set of genes on them. One's from your mom, one's from your dad. But now they're just doubly big. Like they've made a copy of themselves, made a copy of itself. Same thing with these guys. These are still homologous chromosomes, but they've duplicated, okay? And so far, we have not started mitosis or meiosis. Like this is just, after interphase, you now have the 4 chromosomes have duplicated, okay? Now, in mitosis, just to remind you guys, let's just go over mitosis real quick. Here's what happens in mitosis. After your chromosomes have duplicated, basically the gist is, what's gonna happen is they all line up individually like this, and then the sister chromatids get pulled apart to opposite sides of the cell, which are then going to form 2 new daughter cells. So these guys are going to separate, and these guys are going to separate, and these guys are going to separate, and these guys are going to separate to different sides of the cell. And so now you have a set of chromosomes over here and a set of chromosomes over here that are going to then, through cytokinesis, form 2 new cells. So let's imagine there's a cell over here and a cell over here. And basically, this is what we started with at the very beginning. We had 4 chromosomes that were unduplicated. 1 from your mom, 1 from your dad, 1 from your mom, 1 from your dad, a large one, a small one, a small one. The instructions for your eye color, your hair color, and your height, or hair type, and then another set of instructions for your eye color, hair color, and your hair type, and then this is your skin tone and your height, your skin tone and your height. Like, right? That's the same thing as what we started with, but now we have 2 of them. That's mitosis. But now let me rewind and show you what's going to happen in meiosis, how it's different. So again, let's go back to after interphase. So you've done interphase, you've made a copy of all your chromosomes, right? So let's go backwards, rewind. So here's after interphase, you've duplicated each of those 4 chromosomes. We're starting with the same thing here. But in meiosis, you're going to start with the same type of cell, a diploid cell, that has 2 sets of chromosomes, 1 from mom, 1 from dad. They've duplicated because you've done interphase, but here's the big difference. In meiosis, let's start with meiosis I. Here's what's going to happen in meiosis I. The homologous chromosomes, they're going to find each other. They're going to find each other and pair up. they're like, oh my gosh, you have the same genes as me. Let's pair up. And this one's like, oh my gosh, you have the same genes as me. Let's pair up. Oh, maybe they don't want to pair up. There. Now they paired up. So the homologous chromosomes are going to pair up during meiosis I. When we start off with meiosis, in meiosis I, they're going to find each other. The pairs are going to find each other. In mitosis, this did not happen. In mitosis, they leave each other alone. They just all line up individually. Well, in meiosis, they're going to pair up. They're going to find each other, the homologous chromosomes, and they're going to line up together. When they pair up, they form something called a tetrad. A tetrad is this structure here that is two duplicated homologous chromosomes pairing up because they're made of 4 chromatids now. Now there's these 2 sister chromatids and these 2 sister chromatids, a total of 4 all together now. We call that a tetrad, tetrad referring to 4. There's 4 of them. And this is a tetrad too. So those are the duplicated homologous chromosomes pairing up, and they're going to line up in the middle. So now they're lining up in the middle with their buddy. This guy's lining up in the middle with his buddy. And in meiosis I, the gist of what happens is we're going to separate the homologous chromosomes. These guys are going to say goodbye to each other forever. They're never going to see each other again. And these guys are going to say goodbye to each other forever. They're never going to see each other again. And what's going to happen in meiosis I is the cell is going to separate those homologous chromosomes after they line up in the middle with each other and then form 2 new cells. So this is going to, after cytokinesis, this is going to form a cell over here, and this is going to form a cell over here. That's what happens in meiosis I, which I'll show you in the slides in a second, is you're basically just taking the homologous chromosomes and you're separating them into 2 daughter cells. So now, in this cell over here, there's actually only 1 set of chromosomes. There's one of these chromosomes. It's duplicated, but this still counts as 1 chromosome. And this has the instructions for your skin tone and your height. But now there's not another chromosome in here that has the instructions for your skin tone and the height. That's in another cell. Like he's all by himself. Like this chromosome is the only chromosome that has the instructions for your skin tone and your height. And then we have one of these guys who has the instructions for your eye color, and your hair type. And he's the only one now that has those instructions in this cell. Okay? His friend, his buddy, his homolog, his homologous chromosome is in a separate cell, like they've separated. And so now we just have 1 set of each chromosome. 1 set of each chromosome. These are now officially haploid cells. So after meiosis I, when you separate the chromosomes, the homologous chromosomes, you now have 2 daughter cells, each with 1 set of chromosomes. 1 set of chromosomes. Okay? They're duplicated. That's okay, but there's only 1 set. Okay, one of these and one of these, duplicated. One of these and one of these, duplicated. That's fine, but that technically is now haploid. There's only 2 cells, 2 chromosomes in this cell now. In this cell, there's 2 chromosomes. They're duplicated, but this counts as 2. 1, 2. In this cell, there's 1, 2. Okay, duplicated, but that's 2 chromosomes, which we started with 4. So we've just cut the number of chromosomes in half. So now there's 2 and 2 in each of these daughter cells. That's what happens in meiosis I. So now in the slides, let's see if we can cement this into your brain now. If you look here, if we go through these 4 phases of meiosis I, prophase I, metaphase I, anaphase I, telophase I, you're going to see exactly that happen. You're going to see those homologous chromosomes pair up and then separate into 2 daughter cells. There is a little bit more that happens though. There's one really important thing that I did not mention, but in prophase I is when those duplicated chromosomes are going to condense. So they condense, the chromosomes condense. The nuclear envelope is going to go away in prophase I. Just like in mitosis, in prophase, the nucleus goes away, the chromosomes condense. But in prophase I of meiosis, they pair up. Look that they're finding their buddies. Like these guys found each other. These guys found each other. These guys found each other. They're going to pair up. This happens in prophase I. And they're going to form these structures called a tetrad. This is a tetrad. When they pair up, this is a tetrad. This is a tetrad. Now, when they pair up, something magical actually happens that I didn't show you with my beads on my desk. There's something called crossing over that will take place. And crossing over is when the DNA from these homologous chromosomes will start swapping with each other. And so you can kind of see clearly in this picture here, the homologous chromosomes are going to pair up. These are 2 copies of each other. These are 2 copies of each other. And what happens between what we call non-sister chromatids, so non-sister chromatids is one of the sister chromatids from this homologous chromosome and a sister chromatid from this other homologous chromosome, like one from your mom, one from your dad. They're going to start swapping pieces of themselves. So they're going to start swapping DNA with each other, and now you're going to have these recombinant sister chromatids. So now these sister chromatids are genetically different than they used to be. They swapped pieces. You're mixing up your mom and your dad's DNA between them. That's called crossing over. And that happens when the homologous chromosomes pair up during prophase I. Crossing over takes place during prophase I. And so now you're going to have some of these guys now looking a little different because of crossing over. So that's what's going to happen in prophase I. Now, the way that crossing over takes place, like the pieces that are being switched between them, that is actually something that's very random and happens differently almost every time meiosis takes place. So right here in this picture, you guys can see these two homologous chromosomes pair up and they're swapping this piece and this piece. Well, the next time meiosis happens, when they pair up again, let's say meiosis is happening in a different cell, it's always going to be slightly different. They're always going to be swapping different combinations of DNA. It's never going to happen the same way twice. So this is very important. We'll talk about this later. But crossing over creates very unique combinations of DNA between your mom and your dad's DNA that have not existed before. And if we were to rewind and do this all over again, it would be a different pattern. A different pattern of crossing over will take place. It doesn't occur the same way again and again. It's unique every single time. Anyway, that's what happens in prophase I. In prophase I, the chromosomes condense, they pair up, and crossing over takes place, and the nucleus is going away. And again, you have your centrosomes here, which are building these microtubules, moving to opposite sides of the cell. And then in metaphase, what happens is they line up in the middle. But notice what's different here between what we're looking at this picture and in mitosis. Now they're lining up with their homolog. They're as homologous pairs paired up, they're lining up together in the middle of the cell. And you guys can see that crossing over already took place between some of these guys when they were pairing up to begin with in prophase. And now in metaphase I, those homologous chromosomes line up in the middle. Each of these tetrads line up in the middle. And then in anaphase I, remember A for anaphase, A for apart, you're going to pull apart those homologous chromosomes. So in meiosis I, during anaphase, you're pulling apart the homologous chromosomes. This guy and this guy are being pulled away from each other. This guy and this guy pulled away from each other. This guy and this guy pulled away from each other to opposite sides of the cell, and they're going to be packaged soon into two new daughter cells. But those homologous chromosomes will now forever be separated into two different daughter cells. This is different than in mitosis. In mitosis, we were in anaphase of mitosis, they all lined up individually. They were all lining up individually, not together. And we were pulling apart, not the homologous chromosomes, we were pulling apart the sister chromatid. But in this case, we haven't pulled apart the sister chromatid yet. The sister chromatid and this duplicated chromosome, they're still together. They're still together. They're still together, still together. We haven't pulled those apart yet. But what we are doing is pulling apart the homologous chromosomes. And then in telophase I, we start, the chromosomes start to uncondense, and we start rebuilding the nucleus. And then we're going to do cytokinesis, where the cell is now going to, the cytoplasm and the cell is going to now split into two individual cells, each containing one set of homologous chromosomes, or one set of chromosomes, and the homologous chromosomes are now no longer together. And so now you're going to have 3 chromosomes in this cell that's forming and 3 chromosomes in this cell that's forming. So in this picture, we started with 6 chromosomes. If you look up here, we had 6 to begin with. And they paired up and crossing over took place. They all lined up in the middle, 1, 2, 3, 4, 5, 6. And then we pulled them apart and packaged them into 2 different cells. And now there's 3 and 3. One of these, and one of those. One of these, and one of those. These are now haploid cells after meiosis I. They only have one set of chromosomes, So that's basically what happens in meiosis I. And then after meiosis I, you're going to move on to meiosis II. So then each of these cells is going to divide again in meiosis II. Now, meiosis II is very much the same as mitosis, actually. Basically, now they're going to line up by themselves because they don't have a partner to line up with now. And this is when we're going to pull apart the sister chromatids into 2 new daughter cells. And so here's, just to remind you, here's one last picture reminding you what happened in meiosis I. You start with a diploid cell that had 6 chromosomes. They paired up, crossing over took place, they line up in the middle, the homologous chromosomes get pulled apart from each other and packaged into 2 new cells that are now haploid, 3 chromosomes. Now in meiosis II, We're going to now continue on to Meiosis II. Between Meiosis I and Meiosis II, there's no interphase again. So interphase, we're going to reduplicate the chromosomes or anything like that. We're just going to pick up exactly where we left off. And so you have these two cells here that each contain 3 chromosomes that are duplicated. And now in Meiosis II, here's the gist. They're just going to line up individually now because they don't have their homologous pair is gone. It's in a separate cell now. So now they just line up individually by themselves. And this is when we're going to pull apart the sister chromatid and package them into 2 new cells. And to show you that visually quickly on my desk, this is where we ended up with my example. So in my example, there was 4 chromosomes. And if you guys remember, they paired up, right? This is what happened in Meiosis I. They paired up, and the crossing over took place. Here, I'll even show crossing over taking place. I'll switch pieces of them. So like this piece and this piece, just to show you. Sure, there's some crossing over taking place here. Do that right there. So they switch pieces, and then over here, maybe these guys switch pieces. Okay. And then crossing over going to happen in lots of different places, lots of different times, but just keep it simple. So they paired up in Prophase I. This was Prophase I. They paired up. Crossing over takes place. Crossing over takes place. Then they line up in the middle, and we pull them apart, the homologous chromosomes. And then we have 2 new cells, this cell and this cell, that are forming. Okay, that's the end of Meiosis I. These are now haploid cells. In Meiosis II, in each of these cells, what happens is now in this cell over here, these guys are going to now line up individually by themselves because they don't have a partner now. They line up in the middle of the cell. And now we're going to separate the sister chromatids and package those into 2 separate cells. So now that's going to be packaged together, and this is going to be packaged together. So imagine this is now a cell that has one of those sister chromatids and one of these, and this is now a cell that has one of those and one of these. And the same thing with these guys, and this cell that was over here, these guys are going to line up in the middle, and they're going to separate and separate. And so now you're going to have a cell right here that formed, a daughter cell here, a daughter cell here, a daughter cell here, a daughter cell here. So now you're going to have 4 cells. And each of those cells is now going to have a copy of each type of chromosome. But they're still haploid. The only thing different now about each of these 4 daughter cells is now the chromosomes are unduplicated. So we just took those duplicated chromosomes and we unduplicated, but now in this cell here, it has one of these chromosomes, which has the instructions for your skin tone and your height, right? And then on this chromosome, there's the instructions for your eye color, your hair color, and your hair type, right? Like it has a complete set of chromosomes, but just one set of chromosomes, okay? And it has all the genes on it. Same thing with this cell. It has the instructions for your skin tone and your height. It has the instructions for your eye color, your hair color, and your skin tone. It has one of these and one of those. Just like this guy has one of these and one of those. This guy has one of these and one of those. This guy has one of these and one of those. So each of these 4 daughter cells has one set of chromosomes. They're haploid, but those chromosomes are now unduplicated. So we just, in Meiosis II, we basically just took duplicated chromosomes and we made them unduplicated. So now we end with 2 unduplicated chromosomes in each cell. And so these are the final products of meiosis. So after Meiosis II, you now have your 4 final daughter cells, each of them having one set of unduplicated chromosomes, okay? These would be now your sperm or your egg cells that you just formed, okay? And if you notice, all of them are technically genetically different. Like none of these cells are completely identical with each other. Like in this fake example, the simplified example, like this one has a large one from your dad and a small one from your dad, but this one has a large one from your dad and a small one from your dad, but pieces of them have swapped with your mom. So these guys are now genetically different than these guys over here. It's not the exact same DNA, okay? And so we swapped some of the DNA. And then same thing with these chromosomes here, like this one and this one. This is the small one from your mom, the large one from your mom. But this one's the small one from your mom and the large one from your mom, but pieces of the DNA had been swapped during crossing over. So this is technically a different combination of DNA than over here, which is a different combination of DNA than here and a different combination of DNA here. So we ended up with creating 4 genetically distinct daughter cells that are each haploid, which is another difference between this and mitosis. In mitosis, the daughter cells are identical. It's the exact same thing. The daughter cells end up with the exact same DNA, the exact same copy of each chromosome. These guys, things got mixed up a little bit. And so, anyway, that's what's going to happen in Meiosis II. So if you guys look at these slides now, in Meiosis II, basically you're going to have another round of prophase, metaphase, anaphase, telophase. And so basically in this, I'll show you this picture here in prophase. So here's the two cells that we formed after meiosis I, each having 3 duplicated chromosomes, 3 duplicated chromosomes. In this example, we're looking at 3 and 3. And so in prophase II, the chromosomes start to condense again and the nucleus goes away. And then in metaphase II, they line up in the middle. They're all lining up individually. And then in anaphase II, we pull apart the sister chromatid, not the homologous chromosomes. Now we're pulling apart the sister chromatid, pulling them apart to opposite sides of the cell. And then we're finally packaging them into 2 daughter cells during telophase II and cytokinesis. So that's what happens in meiosis II. You're just taking those chromosomes, lining up them up in the middle and separating the sister chromatids. And so now you have 2 cells forming up here and 2 cells forming here for a total of 4 cells. Okay. And so just to really reemphasize these differences between mitosis and meiosis. In mitosis, we weren't changing the number of chromosomes. So here's a picture of mitosis. We're starting with 6 chromosomes. In this example, in this picture, you start with 6, 3 from your mom, 3 from your dad. Each of them duplicate, right, during interphase. And then once they duplicate during interphase, you can start mitosis, and they line up in the middle, and then you just separate the sister chromatid. Separate those 2 copies, those 2 copies, those 2 copies, and package them into daughter cells. So then you end up with, again, 2 cells that have 6 chromosomes, 2 sets of 3, 3 from your mom, 3 from your dad, 3 from your mom, 3 from your dad, which is exactly what you had to begin with. So you just took a diploid cell that had 2 sets of chromosomes, 3 from mom, 3 from dad, and you ended up making 2 new cells that each have 6 chromosomes, 2 sets of 3, 2 sets of 3, 2 sets of 3, 2 sets of 3. Okay, that's what's happening in mitosis. It's pretty simple now. Now, meiosis, things are a little bit messier. In meiosis, which is this picture here, you're going to reduce the number of chromosomes. So you're going to start, let's say we're starting with the exact same cell that we started with mitosis with in the last picture. You're starting with 6 chromosomes, 3 from your mom, 3 from your dad. So you have 2 sets of 3. Those chromosomes are going to duplicate, just like in mitosis. They duplicate during interphase. Before you start mitosis, before you start meiosis, they duplicate. And then here's what's different, though. They're going to pair up with their homologs. The homologous chromosomes are going to find each other and pair up, And then the crossing over is going to take place between them at these areas called chiasma. Chiasma are these regions where they're overlapping and crossing over is going to take place. And so they're pairing up and crossing over is taking place. And then they're going to line up in the middle with their partner because they paired up. And in meiosis I, we're going to separate the homologous chromosomes. So these guys are going to separate one over here. Same thing with these guys and these guys. So you're going to end up with, after meiosis I, you're going to end up with 2 daughter cells that now have only 3 chromosomes that are still duplicated, but there's only 3 now. The homologous chromosomes have been separated. And then in meiosis II, those 3 chromosomes that are duplicated are going to line up in the middle, and we're going to separate the sister chromatid. So then we finally separate those duplicated chromosomes, now become back to normal unduplicated chromosomes because we're going to separate those 2 copies, the sister chromatids. And so then you end up with 4 final daughter cells that each have 3 unduplicated chromosomes, which we started with 6 unduplicated chromosomes in the original cell. This was a diploid cell. It had 2 sets of chromosomes, 3 from mom, 3 from dad. Now these guys only have 1 set of chromosomes each. They have 3 chromosomes, only 1 set each. This one has one of these, and one of those. And so does this guy, and so does this guy, and so does this guy. And they're all genetically different. They're not identical at the end of meiosis. They're all different from each other, these daughter cells, because of crossing over happening and the way they lined up and were separated. It created some unique cells here. But they are all haploid, and they all have 1 complete set of chromosomes. So they have 1 of each type of chromosome. 1 of each type of chromosome. But there's not a second set, okay? So that's the detailed gist of what's going on in meiosis and how that's different than mitosis. There are 3 events that are unique to meiosis that you guys should hopefully already be noticing because I've been saying them multiple times. One difference between mitosis and meiosis that was pretty obvious was the fact that in prophase I of meiosis, the homologous chromosomes pair up and crossing over takes place. Never does that happen in mitosis. In mitosis, the homologous chromosomes never pair up. There's no crossing over. That doesn't happen in mitosis. Also, in meiosis and metaphase I, the homologous chromosomes line up together in pairs, which in mitosis, they don't line up together in pairs. And then lastly, in anaphase I, of meiosis, the homologous chromosomes are being separated into two daughter cells. And never in mitosis doesn't do that. In mitosis, we never separate the homologous chromosomes and package them into sister chromatids, into daughter cells. In mitosis, all we did in mitosis, just to go back to this picture, is they lined up not with their buddy, and we just separated the sister chromatids, but they still end up with their buddy. Like there's still homologous chromosomes in here and in here. Okay. Whereas at the end of meiosis, there's no longer homologous chromosomes. Their buddies are now all in separate cells. But anyway, this we'll practice going over this a lot, and we'll draw it out multiple times together in class. And so at first it's always kind of confusing, and you guys mix things up a lot. But we'll practice a lot in class, so don't worry. But try your best you can to start pinning down what's happening in meiosis, at least the overall gist of how these chromosomes are pairing up and then separating and then lining up again and separating again. First, the homologous pairs separate, and then the sister chromatids separate. The big idea here is that in meiosis, you're starting with a diploid cell, like this guy right here, but you're ending with haploid cells that are all genetically different from each other, that only have one set of chromosomes, one set. Whereas in mitosis, you were starting with a diploid cell that had two sets of chromosomes, and then you end up with two diploid daughter cells that also have two sets of chromosomes that are exactly the same as each other and the same as the starting cell. Like in mitosis, all you're doing is you're just making copies of the cell that you started with, okay? And all you had to do was duplicate the chromosomes, line them up, and then separate those copies. Where in meiosis, it's a little bit more tricky what's going on here. I hope that kind of makes a good amount of sense so far starting off. Go ahead and practice these questions in your guys' notes. And like I said, in class, we'll try to make this all very solidified in your guys' brain as you think about this. But thanks for watching. I'll see you guys later.